This is the second in a series of video tutorials having to do with SEM imaging involving assessment of enhanced irrigation devices. This tutorial will discuss biomineralization of dentin in the hopes that understanding that process more completely will help with the interpretation of SEM images. By understanding the mineralization process, one can develop a better sense of what happens when dentin is demineralized, as partial demineralization is what happens in both specimen preparation procedures as well as in our endodontic procedures. I'd like to do this in two steps. The first step is to review the biological basis for mineralization and to gain a better understanding of how these amazing structures are created. We will then discuss some of the issues around specimen preparation and how those procedures can affect imaging. This flow sheet describes our path going forward. We'll try to do this one step at a time. After we've taken this journey, we will be in much better place to evaluate the control cases before moving on to the enhanced irrigation cases and the more complex imaging analysis involved. In image analysis, the first thing to do is to pay attention to how your specimen is imaged. By that I mean how your view is oriented. After understanding this, then we'll discuss dentin morphology and the odontoblast itself. In the third video, we will cover the organic chemistry of collagen and discuss in some detail the process of mineralization and the role of non-collagenous proteins that they play in that process. So typically, when teeth are split the way we are doing, you're looking at the axial wall. And it's so very important for you to have a conception of what it is you're actually looking at and the angle at which you're looking at it. If you look at this histological picture, what you are actually seeing here is all the cells of the predentin and pulpal tissue that gets compressed up against the mineralization front, which you see in the darker pink here. So you are actually looking at that border where the dark pink is, which we refer to as the mineralization front. And all of the cells, as well as the non-calcified predentin, that are compressed up into the mass there. So that is what you're looking at. If you look at this TEM, all of these cells are compressed into the mineralization front, which is here. And where that arrow is, that's the start of the predentin. But you can see some mineralization taking place in the predentin, as seen by this arrow, and now outlined in red. Notice how irregular it is. In both conventional and enhanced irrigation procedures, much of the predentin is lost. Indeed, if it hasn't been lost in the specimen preparation procedures. So when you see an image like this, you want to think about the anatomical orientation and view the image in the context of all the things that could possibly affect the specimen. In this telluridine blue stained specimen, you see some of the calcification in the predentin, but most of what you see is the compressed pulpal tissue up against the mineralization front of the calcified dentin. So now when you look at an image like this, hopefully you have a better context and can identify the partial calcification of the predentin, the underlying mineralized dentin, and some of the collagenous matrix of the predentin. See how easy that was? Now when we talk about the morphology of dentin, we're really talking about the functional unit of dentin, which is the odontoblast. Later on in the next video, we'll talk a little bit about collagen biology and the entire biomineralization process that will help you understand these images much better. Now, when you look at the early microscopic studies on dentin, they knew there was a difference between dentin in different locations and even within the same location. And it's very interesting how they actually figured this out. And in the MIE Google group, I have included some of these earlier PDFs, which you can access in the mineralization folder. A lot of this was figured out from the SEM and TEM imaging. In the imaging, it was clear there was a difference in texture between the vast body of dentin, the intertubular dentin, and the dentin immediately surrounding the Tomes process, the odontoblastic process. Notice the much finer texture to the peritubular dentin than the intertubular dentin. And it turns out that the peritubular dentin is much more calcified, and it has no collagen fibers in it at all. It is pure calcified tissue formed from the non-collagenous proteoglycans secreted by the odontoblast. It is also demineralized much faster than intertubular dentin, a fact important in dental bonding, as you probably already know. In the early TEM work, the peritubular dentin was clear, as the proteoglycans which made up the structure are removed during specimen preparation procedures. This diagrammatic example may help you conceptualize what you are seeing when you are looking at the images. From the adhesive dentistry literature, when you look at longitudinal sections, you have to become sensitive to what the peritubular dentin actually looks like, because it is so much more calcified and non-collagenous. In this picture, the dentin, which has been etched with phosphoric acid, you can see some smear layer still present on the surface, the asterisk, and immediately below that, also notice that the peritubular dentin has been demineralized deeper than that of the intertubular dentin, and that the collagen fibers of the intertubular dentin are exposed. So when you look at an image like this, you want to become sensitive to what the subtle changes that decalcification brings to the table. Here I have labeled the areas to help you become sensitive to the small differences. Again, another labeled image that helps draw the distinctions. You will need this refined visual scrutiny to understand what enhanced irrigation actually does to dentin. Now, when you look at the peritubular dentin under the electron microscope and process it in such a way as to preserve the non-collagenous proteins, you get a very different picture of the peritubular dentin. The peritubular dentin actually does have a structure. It's just not a collagenous structure. There is a membranous sheath that separates the intertubular dentin from the peritubular dentin and acts as a protective covering of the odontoblastic process for the entire length of the dentinal tubule. This sheath is called the lamina limitens, and when care is taken to preserve the proteoglycans and glycosaminoglycans, you can start to see how complex this structure is. These are the proteins that actually control the mineralization process in dentin. Now, what's so special about the odontoblast? We refer to them as ectomesenchymal tissue because they derive from neural crest tissue, the neural crest from the first brachial arch. They have gap junctions, just like epithelial cells, but most importantly, they are post-mitotic cells. In other words, this one cell lives for the entire life of the pulp, so one cell can live for 100 years without dividing. It's actually quite incredible. Like neural tissue, they don't replenish themselves by dividing. Instead, they repair themselves. The odontoblast also produces all the collagen and all the non-collagenous proteins needed to produce dentin and mineralization. The odontoblast is a very polarized cell, and it changes its appearance as it ages and also where it is located in the pulp. As I mentioned, it has the ability to repair itself, and TEM imaging of it demonstrates quite dramatic phagosomes, organelles known to do housekeeping, maintenance, and repair. The odontoblast makes many non-collagenous proteins, and these are proteoglycans and aminoglycosamine glycans. And you're going to see how important these are in the calcification of dentin. When we talk about dentin mineralization, we will be discussing these proteins in some detail and how they affect imaging and are affected by specimen preparation. So now when you look at this, you are able to look at this with some context and as far as the angle that you're looking at it and what is likely to actually be present. Next, we're going to look at collagen and biomineralization and see how they affect imaging.